Hello, everybody. Welcome to Chapter 9. We're into microbial growth. So we're going to be talking in this chapter about how we grow microbes and how we study microbial growth. So when we talk about microbial growth, microorganisms or bacteria grow and replicate via a process known as binary fission, in which one cell divides, producing two identical copies of the original parent cell. Each of those cells is known as a daughter cell, and they are genetically identical. So the generation time then is the amount of time it takes bacteria to go through a binary fission. So from one parent cell to producing two daughter cells, that would be one generation time. It's also known as the doubling time of the population because bacteria, they grow whenever they replicate, they double their entire population. So one cell produces two cells. Two cells will produce four cells. Four cells will produce eight cells. Eight cells will produce 16 cells, then 32, then 64, then 128, then 256, and it just goes on and on and on. I think I did, sorry. All right, so bacteria and archaea grow via binary fission, and they grow exponentially, which means they grow at a rate in which they double the population every time. Depending on the doubling time, you can kind of learn about the length of incubation for certain diseases. Because if you have a pathogen that has a short doubling time, that means the incubation period is going to be much shorter as well. If you have a pathogen that has a long doubling time, then you're going to have a longer incubation period. But that also means that you can spread the disease further because people can move from one population to a totally different population with the same disease before they actually start showing symptoms of the disease. And so this exponential growth rate actually has some important consequences medically. If you start with a certain number of cells in some type of food, let's say, that number of cells can double or can become problematic if they have time to double for a certain period of time. And so I always think about picnics, and there's always people that no, I think everybody understands what I'm saying, but a lot of people actually are more, what is the word? They are more cautious of pathogens than those of us who were growing up in the 80s. And so when you went for a picnic, people would, cook out. People would cook food. They'd bring potato salad. They'd have macaroni salad, other, cookies and cakes and other things that would be sitting out. And the food would just be left open, and you just go in and you grab what you want when you want during that period of, 3 or 4 hours that you're hanging out at the park, having a big get together with a bunch of people. So that's great and it's super fun. But for bacteria, this is a great time for them to grow in your food. So if you have like a potato salad that is supposed to be refrigerated, you pull it out and you leave it sit for 4 hours in the on a picnic table, whether it's closed or opened, the environment is closed or open, you're not keeping it in that cool temperature anymore, and bacteria are going to grow. And so you might have 10 cells that were in that potato salad initially. And if the cells are able to grow within a 4-hour period, you can change from 10 cells to 40,000 cells or more, depending on how fast the doubling time is. So let's look at that. So here we have the growth calculation. Growth can be calculated using this equation, n sub t, which is the number of cells in a population at some given time t, okay? So this would be like, this is the number of cells in the potato salad when you checked it, and there was 40,000, okay? Equals and zero, or n sub zero. This is the original number of cells. So this would be that 10 cells were the original number of cells that were in the potato salad. And then 2 to the nth, the little n, is the number of divisions. So say you have some pathogen that doubles every 20 minutes, then in a 4-hour period, you're going to have 12 doubling times, right? And you can figure that out by taking 4 hours, multiply that by 60, because that's the number of minutes, and then divide that by the 20-minute period. And that will give you a 12. So 2 to the 12th. So down here I have 10 cells in the original population. And then here I have your your nth number is 12. And so we're trying to figure out this number, which I gave you before, but n sub 0 times 2 to the 10th to the nth is 10 times 2 to the 12th, which is 10 times 4,096, which gives you 40,960 pathogens in that potato salad. So it can really become very potentially damaging. Different bacteria have different generation times, and different bacteria have different virulence. And so if you have a higher virulence, then you're more likely to cause disease with fewer cells. So if you have a high virulence and you only need, like, say 100 cells, maybe you started with 1 cell initially, but now you have, after 4 hours, you have more than 100 cells. Now you can cause the disease. Make sense? So here is a question for you to figure out. I'm going to actually give you the answer in just a few seconds, but I'll let you guys think about it. So pause the video and try and figure this out before we go over and actually get the answer, okay? All right, so now let's go over how we do this. So remember, n sub t equals n sub 0 times 2 to the nth. These are the things we gave you. So we have 2,600,000 cells. We don't know what n sub 0 is in this case, so we know the nt is this. We have 6 divisions, so the divisions are every 40 minutes. There's 240 minutes, so you divide this and you get 6 divisions. So we have 2,600,000 equals n sub 0 times 2 to the 6th. If you take 2 to the 6th, you get 64. So then we can divide both sides by 64, and we end up getting n sub 0 equals 40,625 cells in the initial population. So you will probably have at least one question. Very likely you'll have a couple questions on your exam. So for those of you who love math, you're welcome. If you don't love math, just remember there's only going to be a few questions about this. But you do want to understand the importance. This is why it is important when you're getting a sample, be it a sputum sample, a blood sample, a urine sample from a patient, that it's tested at a relatively rapid rate. If you let it sit for hours, potentially the bacteria can grow, and then you don't know if there is an actual infection, because in urine, there's a certain amount of bacteria that are allowed to be in the urine before we even consider it a disease, before we consider the patient having a disease, because there's bacteria that are in the urethra. There's bacteria that come out with urine that weren't actually in the bladder. And because of that, we know that there's always going to be some bacteria with the urine. That being said, the amount of bacteria will determine if you actually have an infection or not. If it's above, and I think it's above 200,000, then you have to 200,000 colony forming units, which means 200,000 individual cells that can actually grow and form colonies. Then it's considered potential infection. If it's below 200,000, I think that's the number, then it's not considered an infection. So that's a lot of bacteria that you can have in your urine before an infection even is considered. I'm going to stop this video here because this is a 10-minute video, and we'll start the next video in a little bit. Bye.